It's still a pleasure to be here to recognize and learn from some of our most recently minted distinguished university professors. I'd like to give a special welcome to students who came here today as well and express my personal congratulations to today's honorees, John Carruthers, Anna Sukfong-Luk, and Scott Page. Dean Michael Solomon will have more details about these outstanding professors during their more formal introductions. A university can only be as great as its faculty, and today's honorees are, of course, a most impressive group. Past Distinguished University Professorship recipients have included some of our world's most impactful individuals and scholars. Thomas Francis Jr., James Jackson, Patricia Gurin, Shirley Verrett, and Gerard Moreau, to name just a few. Our regents established the DUPs in 1947 to recognize our very best senior faculty members. They honor exceptional scholarly and/or creative achievements, national and international reputations for academic excellence, and superior records of teaching, mentoring, and service. A great feature of the DUPs is that the faculty members get to name their professorship after a person of distinction in their general field of interest. In your program, you could see whom our today's honorees have selected. I've always felt that it's essential that we take the time to honor academic excellence, the faculty who truly do amazing work, those whose passion for scholarship, research, and creativity inspire others, and those who've made outstanding contributions to their discipline. Last evening at the President's at My Home, we honored 40 of our faculty colleagues who this year were elected to the major academies, the National Academies, Sloan Foundation Fellow, a MacArthur Award recipient, AAAS Fellows, American Academy of Arts and Sciences. 40 of our colleagues received that kind of honor this year. The broad excellence of the Michigan faculty helps to set our university apart in all of higher education. For generations, our professors have been renowned for creating new knowledge and asking the questions that are redefining their disciplines. They advance our commitment to superb teaching and ensure that our students participate in discovery at the highest levels. These qualities of our great research university are essential to our society. They allow us to fulfill our obligation to the public we serve and to drive the cadence of human progress. As the philosopher and U of M professor John Dewey said, knowledge authorized by inquiry is the only kind that is entitled to be called knowledge instead of being mere opinion or guesswork or dogma. I again congratulate today's distinguished university professors. And I thank all of our colleagues for your commitment to research, scholarship, creative work, and to preparing the next generation of leaders and scholars. So thanks to all of you. I'd like to invite up to the stage Dean Michael Solomon to do introduction.
Thank you, President Schlichtel. To begin, I'm very pleased to introduce Professor John Carruthers, trained gastroenterologist and physician scientist who focuses his research in the area of hereditary colon cancer genetics and colon cancer disparities as they relate to both genetics and outcomes. Dr. Carruthers is recipient of numerous federal research grants and maintains an active research lab while also serving as chair of the Department of Internal Medicine, which is home to over 900 instructional, research, and clinical faculty members. After receiving his undergraduate and medical degrees at Wayne State University, Dr. Carruthers trained at Massachusetts General Hospital. He completed a fellowship here at Michigan and then took a faculty position at the University of California, San Diego. It is our very good fortune to have been able to recruit him back to Ann Arbor. An elected member of the National Academy of Medicine and the American Academy of Arts and Sciences, Dr. Carruthers has served on the congressionally created National Commission for Digestive Diseases and has been honored by the Association of Professors of Medicine and the American Association for Cancer Research. We are proud to name Dr. Carruthers the C. Richard Bolin Distinguished University Professor of Internal Medicine and Human Genetics. Welcome.
Thank you, Dean Solomon. I want to thank the university, President Schlichtel, the entire University of Michigan for this wonderful honor. So I named this honor after C. Richard Bolin, who was on the faculty here at the University of Michigan from 1984 to 1995. He's a domer. Went to medical school at Yale, but then served in the Indian Health Service, and he, like me, was a gastroenterologist and a physician scientist, having published over 300 papers. And I'll tell you a little bit about this, he coined the term Lynch syndrome, which is the most common hereditary cancer syndrome known to man. He was also president of our large GI organization, the American Gastroenterologic Association, and he won the Beaumont Prize and the Freedom Wall Medal, which is their highest award, and he was a member of the Association of American Physicians. I just want to say to the students in the audience what lessons I learned from that mentorship. Follow your passion. Dr. Bolin's family was riddled with cancer, and he wrote a book called Cancer Family. So this is known in the literature, but it turns out he was the best person because of his motivation to study cancer biology and genetics with. He also mentioned me on writing. I was not the best writer, but writing is a very important part of getting grants and publishing papers. And at Notre Dame, his major was English. He told me about do's and don'ts during career advancement, willing to let go and let me fly on my own, and he's still a friend and mentor today. So he taught me, along with several other people, to emulate role models, work hard and persevere, be true and honest to yourself, surround yourself with great social support, and train the next generation. What do I do now? And you heard a little bit about this, so I won't spend any time. I'm a clinician. I see patients that are high risk for colon cancer. I do research, as Dean Solomon mentioned, in the field of colorectal cancer. I do educate some undergrads, largely on the research realm, but students, graduate students, residents, and fellows, and give talks and CME talks nationally and internationally. As mentioned, I chair a department, and then I've been involved in national organizations, again, emulating my mentor. So what do? I work in the field of colorectal cancer, and cancer of the colon is also termed the large intestine, and generally, colon cancer develops by a pathway in which you develop polyps, we call it adenomas or adenomatous polyps. And here are some endoscopic examples of that, some of which have turned into cancer. In the United States, it's estimated that this year, there'll be nearly 150,000 cases of cancer. That is the U of M Stadium and another half of the U of M Stadium. And about half of the stadium will die from colorectal cancer this year. It's the second most common cancer incidence between men and women. Now, one of the pathways for cancer goes through a repair mechanism. So we have cells that have nuclei in them, and there's about 5 different repair mechanisms that happen in those nuclei. Turns out one of them, DNA mismatch repair, is the one associated with colorectal cancer. There's base excision repair, there's nucleotide excision repair, there's DNA mismatch repair, which I'm going to talk about today, and there's also homologous recombination repair and non-homologous end-joining repair. DNA mismatch repair, actually, after let's see, does this thing have a pointer? Yes, it does. So we have a template and we have 2 strands of DNA, and occasionally when that DNA is replicated, a mistake happens. And generally our polymerases can find those mistakes, but occasionally those mistakes are baked into after the polymerase has done its work inside the cell. So we have this DNA mismatch repair system that can recognize mispairs, in this case written as a CT, because C normally binds with G, and this pair of proteins, we call MSH2 and 6, can recognize that, call a couple other proteins, signal in that for that piece of DNA to be repaired. They can also recognize something called microsatellites. Microsatellites are repetitive sequences of DNA. One depicted here, where it's an AAAG or tetranucleotide repeat that can pair on the other strand of a TTTC, and any one of these AAG repeats can match with any of these TTCs, and occasionally they can loop out. That is also recognized by DNA mismatch repair by this MSH2-3 combination to actually eventually lead to excise and repair, put the correct number of those repeats in. This system is evolutionarily conserved. It's found in bacteria, it was later found in yeast. In 1993, when I was in Dr. Boland's lab, it was discovered in humans, is one of the reasons why I study this. And in 2015, Paul Modrich, who discovered this in bacteria, was named with the Nobel Prize in Chemistry. He's at Duke University. There are 5 conditions in which DNA mismatch repair cause human disease. Essentially, all of them lead to colon cancer. The most common is the Lynch syndrome. As I said, Dr. Boland coined that term named after Henry Lynch, who unfortunately he passed away this year at the age of 94 years old. Lynch syndrome is caused by a germline mutation in one of these DNA mismatch repair genes. Of all the colorectal cancers in the United States, about 3% of them are this inherited condition called Lynch syndrome. Patients present at young age, largely get colorectal cancer, but can get cancers of the female reproductive tract, the urinary tract, and can have some other rare skin tumors and brain tumors. And we in the clinic, we do family histories on these patients, but we can also do germline genetic testing for mutations in these genes. There's another inherited form of this called constitutional mismatch repair deficiency. This is a very, very, very rare syndrome, because in this case, 2 Lynch syndromes have to marry, and you inherit 2 bad copies of the gene. This is extremely rare, and patients younger than age 10 can develop with colorectal cancer. There are 3 somatic conditions that can lead to cancer. One is called just sporadic microsatellite unstable colorectal cancer, or CRC. This is seen in about 15% of all colorectal cancers. Its defect is methylation of the promoter of one of those mismatch repair genes. They tend to be slightly older patients, the common age for colorectal cancer in the United States. There's another syndrome called Lynch-like syndrome, because it could involve all the same genes as that inherited condition. However, it's 2 non-inherited things that happen inside the cancer. So 2 hits, biallelic mutations, and that's about 1% of all cancers. And then lastly, which I'm going to talk about at the very end of my talk, there's another form that was just discovered about 10 years ago that involves these tetranucleotide alterations, which is caused by inflammation. And that's been all the rage lately. So how does this work? Well, we can take a normal colon, or you have a normal colon that can develop one of these polyps. In that polyp, you can either start off on a bad foot with have a germline mutation, or develop somatic inactivation, and you inactivate this repair system. That repair system, because of the nature of fixing the mistakes done by the polymerase, when those mistakes aren't fixed, you get a lot of mutations in the cell. And we call that hypermutated DNA. So you get point mutations that can occur, as well as frame shifts at those microsatellite repeats because the length is altered. Most of those microsatellites, 100,000 of them are in our genome. Most of them are non-coding, meaning that they don't code for any protein. However, there's about 400 that are within coding proteins. And interesting, when those things frame shift, they make a truncated protein, and it turns out that is very immunogenic to our immune system and can trigger an immune response. You can get colorectal cancer from this process, but on the outcome or precision medicine side, we've discovered, and I'll show you some evidence for this, that the main chemotherapy used to treat patients who get this is something with flyfluorouracil. We showed that these types of tumors are resistant to that chemotherapy, and because they induce immune response, they're susceptible to immune checkpoint, which is immune modulation of this. And overall, these patients, because of that immune response, typically have a better survival than someone who doesn't have inactivation of this repair system. So let me tell you about the mentorship piece. So the there's about 400 of these genes that have these coding microsites. So I had a trainee named Barbara Jung who actually discovered the most common mutation in our laboratory. This is the activin type 2 receptor. This receptor is completely lost expression as you go from normal to cancer in these colon tumors that have inactivation of mismatch repair. Turns out about 83% of these types of tumors lose this receptor. I'm not going to tell you all about activin, but Barbara, this became her career project. She was a trainee in my lab. She's worked out all the functional aspects of this, and she, in September of 2019, became the chair of medicine at the University of Washington. Back when I was in Rick's lab, as I said, this system in the humans was first described in 1993. So while I was in the lab, we had no idea how it functioned in the human. We knew a little bit about the bacterial and yeast. And so one of my first projects was trying to develop a model to try to study this. We quickly discovered, because of the nature of mismatch repair, that if you interfere with Watson-Crick hydrogen bonding, normally a G binds a C, there's 3 bonds, but if you put a methyl group on one of the binding sites, you can interfere with that Watson-Crick hydrogen bonding, and you can trigger mismatch repair to bind to DNA and study how it works. We discovered through that process that mismatch repair in the human is a post-synthetic repair mechanism, meaning that repairs after the S phase, after DNA is synthesized. And if it can't repair, the cells undergo a G2 cell cycle arrest, preventing mitosis. So if you have mismatch repair proficiency, it will recognize that mistake, but the repair will be attempted, or it'll commit the cell to cell death, but the fidelity of the DNA is maintained, so it's not passed on to progeny cells. But if there's no mismatch repair present, as in many of these human conditions, there's no repair attempted, mitosis is completed, and new point mutations are introduced, which helps drive the process of that cancer formation. Now, in patients, we do not treat colon cancer patients with alkylation drugs. We treat them with 5-fluorouracil-based chemotherapy. And I remember the discussion I had with Dr. Bolin that you should just look at 5-FU. I said, well, it's probably not going to work because that 5 molecule doesn't interfere with Watson-Crick hydrogen bonding. Turns out I was totally wrong. We did the experiments, repeated it 12 times, and found out it did recognize it. And it turns out there's about 3 different ways for a ribonucleic acid to be converted to a deoxyribonucleic acid. And prior to this discovery, it was thought that the main toxicity of 5-FU was through its process in RNA. But through a lot of work, we showed that the DNA mismatch repair system is what recognizes 5-FU in DNA to trigger this. This was initially done by some early studies looking at cell lines in which those who retained mismatch repair were 28-fold more likely to die in the presence of 5-FU compared to cells that did not have this system present. Now, does this translate to patients? So this is a study we published 16 years ago showing that patients who had lost this mismatch repair did not respond, had no benefit with 5-FU. And this led to the NCCN guidelines and ASCO guidelines changing the approach to use of chemotherapy for patients, particularly with stage 2 cancer, not yet stage 3 cancer. We went further to look at the mechanism. So why does mismatch repair how does it even recognize 5-FU when the toxicity is mostly supposed to be by RNA, which mismatch repair binds to DNA? It turns out it does 5-FU, about 10% of 5-FU gets converted into DNA. So Aki Tajima in the lab showed this by doing specific assays, show that it can bind to 5-FU modified DNA. And even with the 2 different recognition complexes of mismatch repair, The predominant one is this MSH2-6 combination. It recognizes it the highest, but there's a secondary recognition by the MSH2-3 combination compared to controls. And these were done by very specific kinetic assays in partnership with Richard Kolodner at the University of California. Now, we also had some criticisms because you're still treating the cells with 5-fluorouracil, and it still gets in the RNA. So Mori Iwazima in the lab came up with this brilliant idea in which we created a plasmid with a single molecule of 5-FU engineered on the plasmid. So you're not treating the cells with 5-fluorouracil, you're engineering it as one single molecule in the plasmid. And when you put this plasmid in, you can see only the proficient cells recognize that and kill the cell. And if you take a mismatch repair deficient, is shown in A and B, the cells are fine, but once you put proficient cells there, you can see the cells are undergoing cell death certainly by 24 hours and certainly into 72 hours, just with an engineered one particular molecule in a plasmid because of its recognition on the plasmid. Now, I mentioned this top pathway already. There's another pathway just discovered about 10 years ago where inflammation can inactivate mismatch repair, okay, and cause frame shifts at these longer tetranucleotide sequences and cause DNA double-strand breaks. And it turns out the prognosis for these patients is just the opposite when you don't have the inflammation. They're 5-FU sensitive, they have poor survival compared to this group, they're higher in African American colorectal cancer patients, and this inflammation induced can be found in non-neoplastic, such as in a condition called ulcerative colitis, which is an inflammatory condition of the large intestine that we see in gastroenterology. So in a paper that's coming out in press, we looked at how these tetranucleotide sequences undergo a mutation in the absence of mismatch repair. I'll just say at high level, most of the ones that are mononucleotide or dinucleotide always shrink in size. They go from an A10 to an A9. But tetranucleotides, they can shrink and expand, but there's a dominance of going back to its normal native length and then shrinking for mutation deletional bias over time. Stephanie Zeng-Roginski in the lab showed that inflammation can, so MSH3, this mismatch repair protein, does not become mutated or epimutated. Inflammation just changes its location in the cell. So showing this is the nucleus of the cytosol, increasing doses of a pro-inflammatory cytokine IL-6, reduces it out of the nucleus, picks it up in the cytosol, and it's very unique to this mismatch repair protein. And we've later discovered that based on the structure of this particular protein, that it can shift between out because it has specific nuclear export signals and a specific nuclear localization signal, unlike the other mismatch repair proteins. And that's what drives its inactivation. Once it moves out of the nucleus, you get 8 to 10 times more mutations at those tetranucleotide sequences, and it's associated in colorectal cancers with higher IL-6 or pro-inflammatory IL-6 compared to those who don't have this tetranucleotide sequence. I'm going to skip this one. This is the data showing it's effective in these types of patients, and this was a study published a few years ago. Lastly, I'll say that in the absence of cancer, because these are inflammatory driven processes, in this case ulcerative colitis, it can also inactivate a repair mechanism. And this was we're the first to show that you can get ongoing mutations even without the presence of neoplasia that can possibly lead to neoplasia in this high-risk condition called ulcerative colitis. I'll just say where we're going in the future with this. We're going to try to determine the mechanism for this shuttling in and out of the nucleus. This is a novel process for this. We're assess expanding the use of these tetranucleotide sequences for larger panels for patient diagnostics. We're also assessing this in non-cancer conditions. We're assessing the role in double-strand breaks, the use of inflammatory drugs to prevent this, and the role of this in young onset cancers, which I didn't touch, but there's an increase in young onset cancers in the United States. Lastly, I'll say I'm a kid from Detroit. I grew up in a family of a sip of 12. This is me here back in 1968 with my parents. My youngest brother was born that year. 6 girls, 6 boys, and somehow we turned out okay. This is us a few years ago. It's hard to get all of us together these days. And then I'll close by my own family. My wife is in the audience here. This is my wife here. We had 25 years of marriage. This is Rick Bowen at some of the meetings. He gave me his permission to use his name for this professorship. He still lives in California. And my 4 daughters, 2 of which have been here at the University of Michigan. And my dog Max, which is my only other male companion at home. So thank you.
We have time for questions, and I believe there's microphones that are going around.
Sure. Dr. Howe.
Thanks very much. I almost understood everything.
20 minutes is hard.
So, do you see differences among different population groups? I mean, you spent a lot of your career studying disparities in healthcare outcomes and in the process of disease. Is this part of the answer, or not?
So, so Dr. Howe's question is, some of these processes, are they different in different populations? So at the germline level, like for Lynch syndrome, this is in every population. There's a few populations that have founder mutations, but we've seen this in all races across the board. In terms of the somatic inactivation, for instance, the EMAST one with the tetranucleotide sequence, we do see that almost twice as common in African Americans, based on some of the studies we've done. And I could have spent a whole hour just on that topic, but the bottom line is many tumors from African Americans, and you might even port this to inflammatory breast cancers and other cancers, are higher in African Americans. Particularly within the large intestine or colon, we see this as well, and they do have more EMAS. And I believe that that's not necessarily a racial or ethnic thing, but it's probably what's based on the local environment. And local environment is determined by the things that you eat, your local microbiome, and all those things. And we're studying that aspect of it and how that can be modulated. And we do have some very exciting data, which we have not published yet, on the use of anti-inflammatories that vary between these populations as well. Dr. Schlichter, I think, oh, your hands are going up now. There's one back there. With diverticulitis? No, I have not. So the question is, have we studied this inflammatory inactivation of DNA mismatch repair, or EMAS, with diverticulitis? The association with diverticulitis and cancer is very, very weak. And so most people with diverticulitis, as an independent thing, don't necessarily, that's not a pathway to drive to cancer. So we have not studied that. I suspect with inflammation you might get some, but that inflammation is usually short-lived. The inflammation I'm talking about is usually repetitive. So we have a constant level of inflammation and tolerance with immune tolerance in our bowel, because that's a huge immune microbiome interaction there. And there are certain things that we eat that can be even more pro-inflammatory. But in an acute infection like a diverticulitis or appendicitis, I wouldn't expect that to change a stem cell to turn into a cancer. This one back there.
Here, and we use immunohistochemistry for the colon cancer for DNA mismatch repair.
Yes.
Do you think we will have a role for MSH3 immunohistochemistry in like ulcerative colitis patients, just to help see who might progress to neoplasia?
So the question is, so there's a pathologist in the audience. The question is, so right now, for particularly screening colorectal cancers, we do population-based screening, particularly at the University of Michigan and several other major hospitals, in which someone comes in with colon cancer and we stain those for those DNA mismatch repair proteins to see who might be a germline carrier, because we want to know those people and actually get to them and their families. So the question is, we do that, is there any application of EMAS, particularly in ulcerative colitis? So we just published that paper in December, so it hasn't reached the policymakers yet. Our paper, which I didn't go into detail, does show that EMAS goes up the longer you have ulcerative colitis. It also, the level increases from ulcerative colitis to ulcerative colitis with dysplasia to ulcerative colitis with cancer. So we do, from the laboratory, from the research aspect, we do view this as a potential biomarker for that risk of possible transformation. It just hasn't been studied in a larger population or a prospective way just yet. So I imagine down the line that it could be. Any other questions?
Any last questions for our speaker? One more? Thanks, John. Thank you, John.
Oh, hi.
Quick question unrelated to the science, but what strategies do you have to keep the level of enthusiasm and inspiration high in a very competitive field? And how do you overcome failure when it happens as an individual or as part of a team?
So the question is, how do I keep enthusiasm high with all the stuff that goes on? And I say, one, I love what I do. I love being here at the University of Michigan, which gives the freedom to do that. The other thing is, I think working with people and colleagues, particularly some of the young people who come up with ideas, keep refreshing you periodically, presenting to other groups, again, festering ideas, bouncing things off. I think those things keep refreshed. And it gives you a pause for thought for how you approach a problem. And I think, and I said this in my second or third slide after my mentorship from Dr. Bolin, having a good support group, being surrounded by loved ones, I think all those things come into play. I'm also a very optimistic person, so.
Please join me and thank you. It is next my pleasure to introduce Professor Anna Sukfang Lok. Dr. Lok's research focuses on the natural history and treatment of hepatitis B and C and liver cancer. She has published more than 500 papers on viral hepatitis and liver diseases, including guidelines for the American Association for the Study of Liver Diseases and the World Health Organization. Dr. Lok has made important contributions to the understanding of factors influencing the progression of hepatitis B and C and the development of new treatments for these diseases. A graduate of the University of Hong Kong Medical School, she completed her medicine training in Hong Kong and her hepatology training in London under Dame Sheila Sherlock. Dr. Lok joined the University of Michigan in 1995 as director of the hepatology program. An accomplished teacher and mentor, Dr. Lok has also served in key administrative roles, including as assistant dean for clinical research in the medical school. Dr. Lok has received many awards throughout her career, including the Distinguished Scientist Award from the Hepatitis B Foundation. She has also served as associate editor of leading journals in her field. We are pleased to name Dr. Lok the Dame Sheila Sherlock Distinguished University Professor of Hepatology and Internal Medicine. Welcome for her Distinguished University Lecture.
Well, thank you very much, Dean Solomon, President Sisho. I'm very honored to receive this distinguished university professorship. First of all, I would like to tell you my story. As you've heard, I was born in Hong Kong, and I went to medical school in Hong Kong and completed my residency there. Then I went to London and received my hepatology fellowship training under the late Dame Sheila Sherlock. For those of you who are not familiar with the British terminology, that means a sir. And after finishing the fellowship training, I returned to Hong Kong and was on faculty at the University of Hong Kong. I was fortunate to rise through the ranks pretty quickly, but perhaps too quickly. So in 1992, the chair of medicine told me to slow down because there was no more room to move up. And recognizing that it was way too early to start my career, I decided to make a very dangerous move across long distance to this country. Some call that academic suicide. And indeed, for a while, when I knock on the door and explore job opportunities, I was repeatedly told, "You're pretty good, but just not good enough for us." And eventually, I landed a position at Tulane University, and 3 years later, I was recruited to University of Michigan, and I've been here since. There are different types of hepatitis, but I would focus on two, hepatitis B and hepatitis C. These viruses can lead to chronic infection and cirrhosis, which means scarring of the liver, and in turn, liver cancer. Hepatitis B is more common globally, whereas hepatitis C is more common in this country. Together, these two viruses result in roughly 1.2 million deaths per year. This is the reason why, in 2015, the World Health Organization declared that we need to eliminate these viruses by 2030, 10 years from now. It's really scary. How are we going to do that? Well, we know that in people not being exposed to the virus, if you encounter the virus, you might develop an acute infection. And for some, that might progress to chronic infection, and in turn, cirrhosis and liver cancer. Now, if we have a vaccine, we can prevent infection. If we have antiviral treatment, where we can block the virus, eliminate the virus, or suppress the virus, those people who are infected would not pass the infection to other people. And in turn, we will also prevent them from dying from the liver disease. Those are possible strategies. But do we have those tools, and do they work? Before I give you the answers, I would like to take you back to what we know about hepatitis B back when I started my fellowship. And I would begin with hepatitis B. Vaccination against hepatitis B just got approved. That was like a few months before I arrived for my fellowship. And there was no antiviral treatment. In fact, we did not have any assay by which we can quantify the amount of circulating virus. Well, molecular biology was just emerging, and we had hybridization assays, not PCR assays, hybridization assays to measure the amount of virus DNA. But those assays were very insensitive. We're beginning to get hints that chronic infection can give rise to liver cancer, but the evidence was very weak, and people were skeptical. Chronic hepatitis infection can span decades. What happened during those few decades? This is what we knew back then. There was an earlier phase in which the early antigen or the E antigen was present, along with detectable DNA and elevated liver enzymes indicating that there's liver injury. And then, later on, the patients enter into a later phase when the early antigen becomes undetectable, as is the DNA, and we call those patients healthy carriers. Well, we did not realize that the assays were so insensitive that if the DNA was less than 1 million units per ml, we would call it negative. And of course, carrying that much virus does not mean that you're healthy. Well, one of the first projects that I was assigned to work on as a fellow was, one of my former colleagues who also trained with Dame Sheila, who's back in Greece, he discovered that, hey, I can detect hepatitis B virus DNA in some of the patients who are e antigen negative. So these are patients who are supposed to be in the later phase with undetectable viral DNA. But he said, I could find it in some of my patients because it challenged the dogma at that time. He was not able to get his paper published. No one believed in him. So he knocked on our door, and my research mentor, Professor Thomas, said, I was a fellow. He said, go salvage that study and get that paper published. So I say, what am I supposed to be doing? So after scratching my head for a while, I said, oh, you know, they don't believe in that Greek assay, but they're going to believe in the London assay. So we get the samples, we test it with our assay, and I throw in some of the samples from the patients that I was studying, and lo and behold, Stefanos was correct. We validated the results. I sent a paper off to a respectable GI journal. It's one of the few papers that got accepted the first time round. Life is so unfair. But what I learned is, when we challenge the dogma, we come up with a new idea. We should expect skepticism. And we pretty much need to have very robust evidence to convince other people. But I also learned that perseverance prevails if we're correct. Over the next 10, 15 years, I, along with many other researchers, meticulously follow cohorts of patients to truly understand what happens during those decades of chronic infection. I'm not going to get into details, but just to show you how different it is compared to the earlier picture that I showed you. It's much more complex, there are multiple phases, and it's very dynamic. There are constant changes in the level of virus DNA as well as the liver enzymes, and that reflects a delicate balance between the virus' innate desire to replicate and make more of itself, and the host immune response trying to control that virus. But what's more interesting also is that not only do patients progress from one phase to the next, but you can actually revert back to an earlier phase. So this makes studying hepatitis B very challenging, but at the same time, fun. I mentioned earlier that at the time when I began my fellowship, there was no treatment for hepatitis B. In fact, there weren't very many drugs that we could test. One of that was interferon, literally meaning interference with viruses. Interferon actually, back then, was mainly used for cancer treatment. We gave it as IV infusion in mega doses, so high doses that the patients experienced horrible side effects. They would be shaking in their beds, the blood pressure would drop to dangerous level. What do? We said, nurses are not supposed to be administering these dangerous drugs. We need a physician. And the physician would sit by the patient's bedside, monitor and manage those side effects. Who would be the physician doing it? Of course, it's a fellow. And that was my job. Now, very quickly we realized, even if this worked, this is too dangerous a treatment. We can't use that. So we methodically tested lower and lower doses to find what is the minimum dose that would retain an antiviral activity. We got down to about 1/10 of the dose, but we were still administering it on a daily basis. Now, for the students here, what does it mean? That means that as a fellow, I was working 7 days a week for several months. So I said, I need to get out of this. So I designed a pilot study to test whether I can take weekends off. And to do so, I said, well, we're going to administer it Monday, Wednesday, and Friday. Now, my hypothesis was, if the antiviral activity was short-lived, the virus would rebound on Monday after 2 days without interferon. Well, to my delight, there was no virus rebound, and the results were published. And the Fry's weekly regimen became adopted worldwide for more than a decade without anyone understanding the rationale. Now, hindsight, the virus assay, the HPV DNA assay at that time was so insensitive that I would not pick up small rebounds, but no one realized. Interferon did not get approved until 1992, and since then we have a long-acting interferon, 6 oral antivirals that were very simple and safe. But while they suppress the virus, they do not eradicate the virus. They were very effective in reducing liver inflammation, even reversing fibrosis, preventing cirrhosis, and reducing the risk of liver cancer, but not completely eliminating it. Because these treatments do not completely get rid of the virus, it means that when you stop treatment, the virus will come back. That creates a dilemma when we treat patients in the clinic. Well, do I put the patients on treatment and then commit them to lifelong treatment? Or do I watch and wait until it really is dangerous and I got to really jump in and treat the patient? To help physicians determine when to treat, when not to treat, how best to manage our patients, we need practice guidelines. And in 2001, I was very fortunate to be invited by the American Association for the Study of Liver Diseases to lead the development of the first guidelines for hepatitis B. And since then, it was updated several times as new knowledge became available. And in 2015, I was involved in the development of the first WHO guidelines for the management of hepatitis B. Now, the availability of antiviral therapy, even though it doesn't completely get rid of the virus, is really a game changer. As a physician witnessing your patients on the brink of death, put on treatment, and then return to functional life is very gratifying. But we still do not completely understand why some patients would remain stable for long periods of time and others become more active and develop progressive liver disease and die very quickly. After many years of petitioning, in 2008, the NIH funded the Hepatitis B Research Network, which included centers in the United States and Canada, and we focused on patients with quiescent disease, not on treatment, monitor them very closely, and we tested if we monitor them closely and only start treatment when the disease starts becoming active, can we prevent bad outcomes? So late last year, we finally were able to analyze the results. And we found that only 2% of the patients developed cirrhosis, liver failure, or liver cancer after 7 years of follow-up. Guess what? We're devastated. Oh, it's a negative study. We were expecting more patients to die and have bad things happen to them because we had all those brilliant ideas to develop predictive models for bad outcomes. And we're caught by surprise. It took us a little while before we realized, oh my God, this is actually good news. This is great for our patients. If we diagnose them, watch them carefully, and only start treatment when the disease becomes active, we are very effective in preventing bad outcomes. It goes to tell you that sometimes our preconceived notion can completely blind us and take some time for us to get out of it. But we really want to have a cure. Hepatitis B, as I told you, is complicated, so the journey is going to be difficult. However, we started a process. We've assembled stakeholders with experts from the American as well as the European Liver Association, regulatory authorities, FDA, EMA, academics, industry partners, and patient representatives. And we try to map a path from discovery to regulatory approval, and how do we define a cure. Well, I'll now switch gears to talk about hepatitis C. And again, take you back to when I started my fellowship. What do we know? Well, the virus had not yet been identified. There was no diagnostic test, no vaccine, no treatment. In fact, there was no name. We call it non-A, non-B hepatitis. We knew that some patients with hepatitis, it's not due to hepatitis A, it's not due to hepatitis B. We had no idea what it is, so we call it not this and not that. It wasn't until 1989 when a virus was discovered, and then there was a diagnostic test for it. Well, once we know that the patients have hepatitis C, we want to treat them. But we had very little knowledge about the virus, its structure, how it multiplies, and so we went back to interferon again. The response was poor. Initially, less than 5% of the patients show any response. As we extended the duration of treatment and added a second drug called ribavirin, we're getting close to 50% response. But these drugs had horrible side effects. So we knew that this isn't going to last. Well, we knew very little about the virus. We know that we want to have designed the drugs, but we're not ready. But what we knew was people were dying from hepatitis C. So once again, NIH stepped up and funded the whole C network. And we were basically driven by desperation. We need to do something to save these patients from dying. Maybe we can't get rid of the virus, but if we use low-dose interferon, which is better tolerated, and we just bring the virus down by a notch, would we slow down disease progression? So after 8 years, we realized that it didn't work. But we're lucky. Negative study got published in New England Journal. And what we, however, did was we learned so much about hepatitis C as well as about liver cancer, because we were very thoughtful in designing the study, what data we collected, and we had a huge repository of DNA, tissue, and blood. From this one single study, more than 130 papers have been published, with the most recent one only a few months ago. So what have we learned? Well, one of the things is we actually were able to define very clearly the incidence rate of liver cancer among Hep C patients in the United States, which turned out to be lower than that in Japan. We were the first to clearly show that while most liver cancers occur in patients with cirrhosis, it can also occur in patients with advanced fibrosis, and this has major implications in who needs liver cancer surveillance. Because the study was so well planned and we collected so much data, we were able to address questions that we had no idea about at the beginning of the study. Well, in the last 5 years, I actually collaborated with multiple biostatisticians to develop novel statistical methods to analyze longitudinal cancer biomarkers in order to facilitate early diagnosis. This led to publications in biometrics and biostatistics journals that I would never read, and please do not ask me to explain this equation. It's just one of those that I took from the paper. Well, interferon, the reason why it doesn't work well, is an off-the-shelf antiviral. But as we understand the virus more, we're able to design drugs that specifically block viral proteins and viral enzymes important for the virus replication. And we call these drugs direct-acting antivirals, or DAAs. We were one of the first to lead this study looking at the combination of DAAs. So this is, again, very interesting because we compared the quadruple therapy, interferon and ribavirin, plus 2 DAAs, with the dual therapy with the 2 DAAs only. Once again, when we initially saw the result, we were excited by the response in the quadruple therapy arm, 9 out of 10. We weren't smart enough to know we actually make history in the other arm that wasn't as good, because we were the first to show that 2 direct-acting antivirals without interferon and ribavirin can actually lead to a cure. The editorial that accompanied it actually knew the importance and tied to it a watershed moment. And as many of you would know, since then, things have progressed very quickly, and by 2015, we can actually cure more than 95% of the patients with a very short duration of oral therapy that's extremely well tolerated. The story of hepatitis C, from discovery of the virus to being able to cure almost 100% of the patients within 25 years, is a miracle in medicine. But we need to have a vaccine if we want to achieve global elimination. The story about hepatitis B is very different. With a safe and effective vaccine for the last 4 decades, we just need to make sure that we have a better delivery system to places in the world where it is needed most. With effective treatment for hepatitis B, but we really need to continue to search for a cure. I'm here today because I've been extremely fortunate. I had great teachers and mentors who have served as role models for me. That began with my teachers at University of Hong Kong. Sadly, Sir David Todd passed away a couple of years ago, but Professor Young, at the age of 90, is still vibrant, still seeing patients, still serving the public. And I was also very fortunate to be mentored by one of the founders of my discipline, hepatology, Dame Sheila Sherlock. This was when it was an alumni reunion when she was retiring. More than 200 former trainees flew back to London for the celebration. Well, I chose Sheila Sherlock for the Distinguished University Professorship, not only because she founded the discipline of hepatology and was my mentor, but she actually founded Michigan Hepatology, because there have only been 2 directors of the Michigan Hepatology program, and she was also the mentor of the late Keith Hanley. I was also extremely fortunate to have met Barry Bloomberg, the discoverer of the hepatitis B virus, for which he received the Nobel Prize. So, in closing, I want to thank everyone for your attendance, and I'm happy to take questions.
I'm fascinated by the connection between viruses, which are contagious diseases, and cancers, which most of us think is a non-contagious. You know, HPV, for example, is linked to cancer, but for HBV, is it a direct link from the virus to the cancer, or is it something less direct, like a weakened immune system?
So for hepatitis B, there are actually 2 paths. One is through inflammation, cell repair, and DNA damage during cell repair. So that is one process. The other pathway is that hepatitis B virus can be integrated into the host genome and can cause some havoc. Now, hepatitis C does not integrate, so it's mostly through inflammation, cell injury, and repeated cycles of cell regeneration.
Thank you very much. That was a wonderful talk for someone like me who doesn't understand a thing about medicine. Are you taking any new patients, by the way?
I have to, otherwise my chair is going to find me.
So you mentioned early in the talk that there is a geographic variation on the incidence of these different hepatitis. Is there any provisional hypotheses about that?
Well, it relates to, in part, risk factors. So different viruses are spread differently. Both hepatitis B and hepatitis C can be spread through sexual contact with blood, secretions, and all that. But with hepatitis B, one of the reasons why it is very common in Asian countries is passage from an infected mother to the newborn baby. Now, it's a chicken and egg thing, which I can spend another day explaining to you about that. But because a lot of mothers, young women are infected, so they pass the infection on to the newborn babies. Newborn babies have a very weak immune system. When they get infected, they tend to develop chronic infection, and then they just keep going from one generation to another. But the story is a lot more complicated than that.
So what's the reason that for Hep C there's treatment, but there's no vaccine, but Hep B there's a vaccine, but no treatment?
Well, hepatitis B, the first vaccine was actually a plasma-derived vaccine. This is a very interesting virus. It has an envelope, but for reasons that we don't completely understand, the virus actually makes more than 1,000-fold more envelope than it needs to coat the virus itself. So these extra empty envelopes circulate in the blood. So the first vaccine was actually getting people infected, do a plasmapheresis, essentially collect the blood, get the plasma, and spin down to get these empty envelopes in order to make the vaccine. Now, later on, obviously, it was replaced by genetic engineering. And with hepatitis C, the reason why we cannot make a vaccine is because it's an RNA virus. It undergoes very rapid mutation. So there are multiple genotypes and variants, and the immune system is always playing catch-up. So as soon as your immune system is able to figure out how to control this, well, the virus has already changed its size and shape. So that's one of the difficulties.
Are there further questions? Maybe if I could add one quickly. Maybe I'll grab a microphone so I can be heard. I'm just wondering if you could speak briefly to the transition from chronic disease to the clinical presentation, about if it's understood how that happens or why it happens at a certain moment as opposed to other moments.
Well, we know that for patients who maintain high-level virus for a very long period of time, they have a high chance to progress to cirrhosis and liver cancer. But that's not the only thing. In fact, all of these viruses, they don't come in one shape and size. There are different genotypes as well, and certain genotypes are more virulent than others. We certainly know that the host immune response play a very important role. And when patients actually have other concurrent disease for which they receive treatment that suppress the immune system, the virus can shoot up 1,000-fold, 1,000,000-fold. So there are multiple factors. And certainly there are other factors like if people drink a lot of alcohol, hopefully we don't serve too much today, that can accelerate liver injury. And nowadays, actually concomitant fatty liver disease because of obesity also accelerate liver injury. So there are multiple factors that come into play.
Please join me in thanking Dr. Locke for her distinguished university lecture. It's now my pleasure to introduce Professor Scott Page, a scholar whose boundary-crossing research focuses on the many ways that diversity plays in complex systems. An invertebrate asker of simple but powerful questions, he explores interests such as how does diversity arise? How does it make a system more productive? How does diversity impact robustness? His work is valued in many disciplines, including political science, management, physics, public health, geography, and engineering. Dr. Page received his undergraduate degree in mathematics here at the University of Michigan and is a native of our state, and has received graduate degrees at Wisconsin and Northwestern. He held faculty positions at UCLA, Caltech, and Iowa before we welcomed him back to campus in 2000. His research on diversity, complexity, collective intelligence, institutions, and the use of models have been supported by the National Science Foundation and the MacArthur Foundation. Dr. Page is a recipient of a Guggenheim Fellowship and is an elected fellow of the American Academy of Arts and Sciences. Dr. Page is a sought-after speaker, including reaching audiences in organizations such as NASA, Google, the IMF, the Federal Reserve System, and Genentech. His online course, Model Thinking, has attracted over 1 million participants. We are proud to name Dr. Page the John Seeley Brown Distinguished University Professor of Complexity, Social Science, and Management. Welcome.
Thanks. It's fabulous to be here. It's humbling to follow John and Anna. Just amazing. And I want to thank particularly Charlie Doring and Nancy Burns, who I know put a lot of effort into making this happen. And I want to start with really what is a common story. I mean, we come from places with names like Dowagiac, Littleville, Ishpeming, Tecumseh every year, right? I'm from a place called Yankee Springs, Michigan. And this place gives us amazing opportunities. And so, Angel talked about giving an uncommon education to the common person. I am that common person. My mom was township clerk. My dad was a football coach. My uncle was a football coach. We ran a little gas station on a lake. There were 10 kids. Between the two families, 5 came here. And my wife and I, Jenna Bednar is a political scientist, we often have this quote that we use by the poet Mary Oliver called, "What will you do with your one wild and precious life?" And the beautiful thing for me, and I think for all of us, from Pewamo, Westphalia, New Lothrop, Montrose, Saginaw, Buena Vista, all these places, is that Michigan allowed us to have these wild and precious lives. So I thank all of you. These numbers up here, 3/21/63, that's my birth date. I was destined to do math. I came back my freshman year for my cousin's wedding, had my first math exam, and I got a 28. Things looked bad. And my house got a house number. Prior to that, I'd been rural Route 3, Middleville, Michigan. In fact, the people in my hall sent notes home, Scott's mom, rural Route 3, Michigan, just to see if they'd arrive, and they did. But I got this house number 2532, and for a moment I thought, uh-oh, this math thing looks bad, right? Because there's nothing interesting about that. But then I quickly realized 2 to the 5th equals 32, and I picked up my orange pencil and I just continued. So one of the things that I think is so important about people coming from places where I come from, the University of Michigan, is that you get exposed to ideas and challenges that you might not have thought of before. And so I took a class with Lem Johnson on Afro-Caribbean literature my junior year, and Lem was a just remarkable person. And we read some work by Maxine Hong Kingston, and if you know Maxine's story, her father was a brilliant man who came to the United States and didn't have the opportunities that we get by coming here. And this is kind of the flip side of the what will you do with your one wild and precious life. And let me just read this. He talks about somebody working in a bike shop, insert hooks in pegboard, down again, up one more time with the bike. The hooks do not meet the frame. If part of the bicycle fits on one hook, the rest of it does not fit on any of the others. Down, I have not found right livelihood. This is not my calling. Owe it a waste of my one and only human life and now time. And so when I think about my experience here, it has just been such an honor and such a privilege to have this opportunity, and I thank this entire institution. And so today, it's huge fun. So this is JSB, John Seeley Brown, the so-called chief of confusion, Michigan PhD, a person with tons of ideas. I asked my son Cooper to make a slide representing John. This is about the best we could do. It's fantastic. He's just a man full of life and full of joy. He has all these honors and awards. He's on Amazon's board. He's on the MacArthur board. He was chief of research at Xerox PARC. Tons and tons of accomplishments. And what I love about John is also what I love about many of my colleagues here at Michigan, like Bob Axelrod, Paul Caron, Carl Simon, is they just love ideas. And John now sort of accumulates things like honorary degrees and stuff like this. And this is a quote he often gives. He says that the greatest thing a human soul ever does in this world is to see something and tell what it saw in a plain way. So what is it that I do with my wild and precious life? It's a strange thing. What I do is I write models. It is an odd thing to do. And I write simple models. And so what I want to do today is I want to just show you and give you a feel for how this gets me up every morning and why I think this is important and why I think it's wonderful that a place like this can support it. So what is a model? This is the famous Harry Beck model of the London Underground. So Harry Beck worked in the signals office of the London Tube Station, and he wrote this map of the London Tube. It's a remarkable map because it doesn't look anything like the geographic thing, right? It's not realistic. So he made this in 1931, and in his spare time kind of passed it around, and eventually in 1933 they decided to publish it. And by 1935 everybody in London had a copy, right? It worked well not because it was accurate, because it captured the essential features of the world. So hang on to this idea of a map. What we do with these models is we tend to think, oh, I write down a model and then I take it to data and I test it. But one of the things we do with models is we just explore the world. We just think. And I want to give sort of two examples of two people who I love dearly. So the idea of this exploring is that we don't care about facts. We just want to kind of figure stuff out. So John Holland one day says this. We're sitting around in a group, Carl Simon, Rick Riolo, Bob Axelrod, and John's like, imagine there's just a bunch of billiard balls, and they're different colors, and they just meet in groups and they bump into one another. And when they meet, they become different colors somehow. What can happen in such a system? And one of the things that John was really interested in is what makes a system complex. So a system's not complex if it's like a basin, so every time it just always goes to the same thing. It's complex if it can be path-dependent, if you can get different outcomes. So I thought about this, and this is kind of like a Jimmy John's problem. Like you go to Jimmy John's and take out your napkin and you think, you gotta be able to figure this out. And so I figured out one thing about this, and I wrote a paper on it called the Rule of Six. It turns out if the number of items plus the size of the groups, the number of colors plus the size of the groups they interact in adds up to 6, you can get complexity. If it's less than 6, no complexity. 2 colors meeting in triples, no complexity, right? 3 colors meeting in pairs, no complexity. 3 colors meeting in triples, complexity. 4 colors meeting in pairs, complexity, right? So it's kind of cute. Not important, maybe, but fun. This is Jenna Bednar, my partner, our complicated lives, I guess. And one day Jenna comes up to me and she said, I'm teaching this class with Ron Engelhardt on culture, and one of the hallmarks of culture is that like there's a consistency to cultures. Like so when you go to France, like the French are kind of all the same, right? If you go to Sweden, the Swedes are all the same. But there's also tremendous sort of within group heterogeneity. And so we have these models of culture. There's some guy named Axelrod that wrote this model of culture. And Axelrod's model of culture, what happens is people coordinate with other people. So we all either shake hands or we bow, we samba, we waltz. There's these things that we do. And Jenna said, imagine if not only coordinating, we also have this desire to be consistent. So Jenna has this wonderful slide on this. Like it'd be really inconsistent for me to like sort of like hug someone, right, and then waltz, or inconsistent for me to bow and then have our national dance be the samba. Right, that just doesn't make sense. So how do you, so she goes, imagine we just had this model where people on the one hand were trying to minimize cognitive dissonance. I want to be coherent, but at the same time I'm trying to coordinate with everybody else. And the simple answer would be, well, you just get everybody would coordinate and be consistent. But it turns out, this is sort of an idea that goes back to Schelling, there's this sort of paradox of aggregation here. The macro behavior is very different than the micro foundations of this. Because there's a little bit of error, a tiny bit of experimentation. What you end up getting is you end up getting an enormous amount of diversity. So if you look at the world, if you're Ron Engelhardt, you spend 30 years traveling the world and saying, wow, there's so much variation within cultures. Why does that happen? It turns out Jenna's answer is it's because everybody's trying to be the same. Right? This is why we write models. Because there's an ecological fallacy. What we see at the macro level isn't necessarily true at the micro level. So let me go to a very specific question. And I want to sort of show you sort of just unpack the process of building this thing. So what role does cognitive diversity play in collective intelligence and groups of people being smarter? And I want to look at a very specific task, which is a task of classification, of binary decisions. So this is a jury trying to decide guilt or innocence. This is the FDA trying to prove a drug. This is the case where there's a right answer. We have a bunch of smart people in the room. Marvin's in the room, right? Mark's in the room. We're all trying to decide. Edie's in the room. We're trying to decide, what should we do? And we want to ask, does diversity make this group better? Now, this goes back, there's an old theorem, the Marquis de Condorcet from 1795, called the Condorcet jury theorem. The Condorcet jury theorem is a mathematical model that says this. There's a binary choice, it's either true or false. Each person gets what he calls a signal, and that signal has some probability p of being correct. It's correct more often than not, right? And then what happens is, we're going to assume that these signals are independent. So the probability that Carl's right is independent of the probability that I'm right, right? And we just kind of want to ask what's going to happen. This independence assumption is kind of sneaking diversity in, right? So the only way that Carl and I are going to be independent is if somehow we see the world differently, if we're getting different signals. And so there's this great theorem, and this theorem basically says that juries are going to be more accurate than individual people. And the larger the jury is, the more accurate it's going to be. And so this is nice, and you can sort of infer from this that, well, we shouldn't have single judges, we should have Supreme Courts, and we shouldn't have dictators, we should have democracies, but that's probably reaching too far. But when you take a model like this, one of the things that they do is they travel. And one place it traveled is to one of Michigan's other common people who came here, named Claude Shannon, who's born in Petoskey, lived in Gaylord, Michigan. And he came here, and then he eventually went to MIT, and he eventually went to Bell Labs. And when you're sending messages through the air, if you're sending little strings of zeros and ones, mistakes can happen. So how do you prevent those mistakes from accumulating? What you do is you make multiple copies. So you send multiple messages, and then you have the messages vote, and then you no longer have errors, right? So this is this wonderful case of sort of a mathematical theorem basically written down for voting that ends up being used for the phone company. But here's the rub. That model kind of sucks. I mean, it's not the case. Like if Ron Sooney and I are sitting trying to decide the guilt or innocence of someone or making a tenure case. We sat on a tenure committee for a long time. It wasn't like we stood there and passively received some signal. We worked. We read these books for hours and hours and then had long discussions about what was true. So it's not the case that we get these complete signals. And so you think, shouldn't I write a more accurate model? But here's why you can't. There's this wonderful, this is from Borges on exactitude in science. One person says, what a useful thing a pocket map is. They've got this, this back map of the tube. It's fantastic. And Meinherr, who's the protagonist, says, that's another thing we've learned from your nation, map making. But we've carried it much further than you. What do you consider the largest map that you would be useful? The person says, it's 6 inches to the mile. mine here says only 6 inches, we very soon got to 6 yards to the mile. Then we tried 100 yards to the mile. And then came to the grandest idea of all. We actually made a map of the country on the scale of a mile to a mile. The perfect model, the perfect map. So have you used it much? Actually, it's never been spread out because the farmers objected and they said it'd cover the whole country and shut out the sunlight. And so now we actually just use the country itself as its own map. And it kind of works okay. So this is the paradox of modeling, right? The model's got to be simple to be of use, but if it's going to be simple, it's going to be wrong. And let's go back to John Holland, and let's go back to this idea of sort of complexity. The information content in a complex systems is large, right? And so no simple model can possibly explain it. So what you want to do is you want to use many models. So most recent work is this book called The Model Thinker, which talks about if you want to understand a complex world, what you want to do is you want to throw an ensemble of models at things. So now I'm going to do something that takes a lot of hubris. I'm going to offer up an alternative to the Condorcet-Jury theorem model. But I'm not going to do it alone. I'm going to do it with Lu Hong, who's my co-author of 28 years. And Lu and I have been co-authors so long that for the first 3 years of our co-authorship, we sent our research back and forth in the mail. So we are a pre-internet collaboration. And I would sort of like live in fear of an envelope coming from Lu because it would always come back with like, this is interesting, here are 7 mistakes. Okay, so Lu's a mathematical economist at Loyola University in Chicago. So the essence of the Condorcet-Jury theorem is randomness, it's uncertainty. But at Michigan, one of our great sort of claims to fame theoretically is this notion of sort of complexity, things that are high dimensional, difficult to evolve, engineer, predict, those sorts of things. We take the details seriously. So what we do is we say this, we say there's some state of the world, this is a simple model of a complex system. Imagine this is the set of all things that could be out there. And there's some of them that are true and some of them that are false. Now, I'm going to have a model of that system, but that model is going to be a simplification. It's not going to get it right. So this may be, Lu, and Lu may have a model as well. So this will be Lu's model, and she's going to say everything over here is true and everything over here is false. And I'm going to have a model, and my model is going to say everything in the top is true, everything in the bottom is false. So we're both going to be right, kind of like the Condorcet-Jury theorem, about 75% of the time. But instead of saying probabilities, I'm actually taking seriously this notion of a state space. Okay, but now I've got a problem because I've got 2 of us, right? I've got Lu and Scott, and so it takes 3 for a jury. So I'm going to bring in Carl Simon. Why? He's a beautiful human being, and because if anybody loves modeling more than I do, it's Carl. Here's Carl's view. Why is this Carl's view? Because Carl is a human being, and human beings, we reason from exemplars, right? So Carl has lived this wonderful life, and he's got all sorts of experiences, and he says, this looks like this experience that was true, and this looks like this other experience was true, and this looks like this other experience that was true. And so he's got a mapping that looks like this, and it's also right about 75% of the time. So what happens when the 3 of us get together and vote? Well, down here in this corner, Lu and I both say false, so that's what we predict. Up there in that corner, Lu and I both say true, so that's what we predict. But then on the off diagonals, Carl kind of breaks the tie. And if you look at how we do collectively, we do better, right? We're probably going to be 85% right. So when you think of this, you can think, wow, this is fantastic. So if we have diversity, we're going to be better. And you think, I've sort of painted a mathematical picture saying, if people have diverse perspectives, then collectively we're going to make more accurate predictions, and that seems wonderful. And then you can think, well, isn't that just kind of what the Condorcet-Jury theorem was doing with probabilities, and didn't you just complicate things? Well, it's not quite true. So Lu and I have a paper from 2009, a decade ago, that says this. It turns out that it's all but impossible to write maps, right, where those maps where Lu looks at this direction, I look at this dimension, such that you actually get probabilistic independence. In fact, the only way you can do it is if the underlying space is n dimensions, and each person looks at all but 1 dimension. So if there were 17 of us, you'd need a 17-dimensional problem, and each person would have to look at 16 different dimensions. So in short, can't happen. So in short, the 2 models just are incommensurate. The one is about probabilities, the other is about complexity, and independence is something that won't be true. So that problematizes this notion of diversity suddenly being good, because the independence as a notion of diversity now doesn't hold in this world. So the Condorcet-Jury theorem is saying accuracy resulting from this independent errors canceling out, and Lu and I are saying, no, maybe accuracy actually comes about from the fact that people have different maps. And diversity is going to be how often people sort of come to, like how often Ron and I disagree, which is only what? 25% of the time, maybe, right? Ron's saying 20, but maybe it was 25. So then here's the question. Is more diversity better? So I'm going to do an example. 5-person jury, each one's right 60% of the time. Each person at 60%, the red line is Condorcet. So if I take Condorcet's model and say, what are the odds that 5 people each right 60% of the time voting, get it right, it's going to be 68%. So what I'm going to do is I'm going to do a thought experiment, and on this axis, I'm going to ramp up diversity. And if I were going to have some beautiful Michigan moment here, as I ramped up diversity, you would just see accuracy go up, right? It'd be beautiful. Doesn't happen. What happens is, you ramp up diversity, good things can happen, bad things can happen. Why is that? Well, look, down here, there's a set of times Carl is right, set of times Lou is right, set of times I'm right. We're going to do really badly if we have a bunch of 5-0 correct votes. If we waste all of our good votes, right, where it's 5-0. We're going to do really well if we pool all our incorrect votes. But this is only for a little bit of diversity. Let's go spinal tap here. Let's crank it all the way to 11, right? When you crank it up and you get a lot of diversity, suddenly you always do better. And in the case of maximal possible diversity, that means every vote's going to be 3-2. If every vote's going to be 3-2 and we're right 60% of the time, we get it right every time. So the story is little diversity is dangerous, a lot of diversity is good. So how do we get rid of this, right? How do we get rid of this thought down here? How do we sort of push past this diversity threshold and get good answers? Well, we can look to computer science. What does computer science say? They have these things called random forests. Random forests say, let's create a bunch of little predictors, right, that look at a bunch of data, and then they vote, right, on what something is. The way random forests get diversity is they do this thing called bagging. They give them different experiences. So the story behind this algorithm is if Carl and I have different life experiences, if Lou and I have different life experiences, then we're going to do better. This makes a lot of sense. How would that work with people? Probably not like this. Right? You probably want people who come from different places. This, by the way, is the Volkswagen board, who is known for many things like cheating on their emissions tests. So this notion of different life experiences leading to more diversity, leading to good outcomes, suddenly has a lot of traction. But there's something deeper here, and I want to touch base with Pat Gurin again, who's been a mentor of me since I came here to Michigan. When we're in that space, and we disagree, and Carl says I see it this way, and Lou says I see it this way, and I say I see it this way, and Ron says he sees it this way, and Jenna says he sees it this way, there's a possibility for a deeper integration, for having what she calls a dialogue across difference. And this leads to something that Robert Aumann, the Nobel Prize winner in economics, called the common knowledge problem. And I call this the green apple problem, and I teach it to my undergraduates. So suppose we have a green apple, and suppose we have 2 children. These are our sons when they were younger and cuter, Cooper and Ori. And we're trying to, if truth be told, they'd rather eat foot-long hot dogs, right? But suppose that we have a green apple, and we're trying to decide if they're going to eat it. Well, one model might be the fruit model. It says it's fruit. They're going to eat it. Another model might be the green model. It's green. No way they're going to eat it. A third model might be it came from Stuckey's. Not going to happen, right? So the vote's going to be 2-1 against. But what Aumann says, and what Pat Gurin would said, is let's talk about it. Let's realize it's a green apple from Stuckey's. Let's have a dialogue, unpack the whole thing, and we'll get it exactly right. That also seems a little bit too convenient, right? It may work for a green apple, but doesn't work in general. So Alain Landemore, who's a political philosopher at Yale, and I thought deeply about this because we thought, like, that just seems too easy, right? That we can sort of like identify to the state of the world and immediately know what's going to happen. And so she wrote a paper that I don't fully understand because it uses words like positive dissensus, but I try. But the basic idea is this. If you have 3 economists, and one is trained as a Keynesian, one is trained by Nancy Stokey like I was, and one is trained by Karl Marx, they have deep, deep understanding. So it's not just green, not green, fruit, non-fruit. These are really, incredibly intense and sort of thoughtful, fine, granular interpretations of the world. And you're going to have these discussions, and you're going to get close. You're going to get better. And the real possibility lies in deep conversations between groups of different people. And I think that's where the promise lies. So does diversity give us better outcomes? I think the answer is, if we have a lot of it, in this case, yes, it absolutely does. And if we have some of it, it does, but only if we deeply engage one another. So I want to end with a quote from my friend Catherine Phillips, who we actually lost this week. And Catherine wrote the ending of my book, of this book I wrote, The Diversity Bonus. And in it, she says this, and it's just a beautiful idea. She says, you should not stop chasing after the benefits of diversity because they're difficult to capture or because some are resistant. Diversity requires us all to work hard and to embrace the change required in ourselves and in others around us. And she's absolutely right. So does diversity make us better? Yes, but it takes work, it takes effort, it takes a commitment, and it's a privilege to be at a place that takes that seriously. Thank you very, very much.
We have microphones available for questions.
Carl, you can ask about mass if you want.
Let me begin. This is the first I've heard about this idea that there can sometimes be a crossover from where a small amount of diversity gives you a certain kind of result and a large kind of diversity. Is there any way to think about where that transition actually occurs in a way that would be useful beyond the particular example that you gave?
So one of the things I think is really fascinating, it's the thing I'm teaching an undergraduate class here at Ross on collective intelligence, is the diversity here is kind of like our maps of the world. Like how are you sort of unpacking things? How are you parsing things? How do you interpret things? And so what really drives, you can think of this in terms of correlation, what really drives the crowd sort of getting it wrong is us making the same kind of mistakes. And so when you think about, so a big question in this space is how do you weight things? And so one of the things we think in democracy is we should weight things equally. But one of the things that comes out of this is that you maybe should weight by ideas as opposed to by people. So suppose 5 of us are voting and John and Anna are in the same lab, have the same model. Well, if we give them 2 votes, we're giving that model 2 votes, right? As opposed to if we give them 1 vote, we're giving that model 1 vote, right? And so as a general rule, weight, and this is one of the sort of ideas from Random Forest, or ensemble learning more generally, is weighting should be done by model as opposed to by number, right? And so that's why diversity becomes so important. I could have a diverse group, but if 6 people are using the same model, I might as well just have 1 person in there, if I've got a group of 9. It's also, one of the things I didn't talk about is there's this fabulous idea called boosting. And the idea behind boosting is when I've got lots of data, what I can do is look at where I was wrong. So before, when I train those trees in computer science, what I do is I treat all the cases the same. Everything gets a weight of 1. But what I could do with boosting is after I see which ones I got wrong, I give those a weight of 3, or a weight of 5, or a weight of 7. And then I retrain new trees to put more weight on the ones I got wrong. And that'll also move me out of that space. But this is a standard idea from some of my business school colleagues here. From business school, analyze, learn, right, and then change things. And so one of the things that comes from this is you want to have diversity on the hard problems, right? And you want to encourage people to sort of think deeper and have deep dialogues on these hard problems. That's where you really get the bang for the buck.
Let me ask you about an application, which is interdisciplinary research teams.
Yeah.
So everybody says that's wonderful, they're diverse, and therefore they can be
Right.
But the problem often is that people from different disciplines can't understand each other.
Right.
They learn from their discipline a bunch of things, but those are often not very much overlapping between a mathematician and a chemist or whatever.
Right.
So how do you overcome the idea that this kind of interdisciplinary diversity, it comes up against the barrier of not being able to have a constructive or even a interpretable conversation?
So Bob's question, just if you can hear, is that like interdisciplinary research, right, has all this promise, but one of the things it suggests is like if there's too much diversity, how can people understand one another? In particular, if Steve Martin has this great line, like those French, they have a different word for everything. right? And the same is sort of true of sociologists, right? You know, they have a different word for everything, and political scientists and economists, and so it becomes difficult to look cross purposes. So the NIH, and actually the Olsons, right, who were here for a long time, helped form this big group with the NIH to look at sort of the performance of team science. And if you read those reports, at first you can think, wow, there's some people saying interdisciplinary is good, and there's some saying it's bad. But if you unpack it, Jonathan Cummings, who's the person who's from Duke, who's basically saying it doesn't work, is saying it doesn't work if you've got people from 11 disciplines at 7 schools. Those projects don't work because for exactly the reason you mentioned, right? You're not capable of having conversations across those groups, right? It's just too big. There's just too many disciplines. And so right now, like Carol Hall, who's the lead person on this, she will say there's an inverted U of diversity of this sort, right? Now the question is, where is the peak of that U? So when you look at a place like the University of Michigan with like 100 departments ranked in the top 10, we'd like to think that we can somehow work to push the peak of that U. And the evidence is that where that peak is has gone from 3 to 4 to 5 to 6 to 7, right? And this is where, again, hearkening back to Pat, are there protocols, are there techniques? So John Seeley Brown, right, who my chair is named after, would say, you need an artifact. Once you have more than like 4 disciplines, you need a physical artifact, because then we can sort of leverage our visual cortices. Like, so if you say, I think this has to be this, I don't know what that means, but if you actually physically build something, I can interpret it within my own lens. And so John was a huge advocate at Xerox PARC of the building of artifacts, but generally, I mean, you're raising what I think is one of the most important questions. How far can we push it? But I think there is a skill set here. I think we can learn to be better at it. Yeah.
Scott, wonderful talk.
Thanks.
There was. Can you hear me?
Now you go. There you go.
There was a paper published in Nature Communications in 2018 about publication and diversity. I don't know if you saw that, but the paper analyzed something like 11 million publications amongst like 18 different disciplines with 5 million authors.
Right.
And they looked at what generate the highest impact. So, and most of us in science worry about how impactful our science is and how it gets quoted. And they analyzed everything from the year of publication, the discipline, all this other stuff. And the number one driver between for the highest impact was the group's diversity, who generate the research work. So when you were mentioning this, you're talking about like someone in a jury room, 2 people interacting. How does this translate to other people citing work that's in a broad? I mean, now I was blown away by that article.
Yeah, so this is work by Brian Uzi, and it's Dashan Wang at Northwestern has just done a sort of a extension of that. So briefly, the idea is this: if you look across every paper ever published, and you say what leads to success, right? What are the drivers? The 2 big things are, one is past citation records of people, and the other one is what they call atypical combinations. So the classic example here is Kahneman and Tversky. You take a bunch of stuff in psychology and a bunch of stuff in economics that's never been cited together in the past. So what Uzi did and Ben Jones did in this paper is they looked at all your citations and they asked, are you citing sets of papers that have never been cited together in the past? And if you have, that's called an atypical combination. And what you see, which is fascinating about this work, is there's a bonus from sort of being someone who's heavily cited that's about this big. And there's a bonus from atypical combinations that's about this big. And if you have both highly cited work and atypical combinations, you get a third bonus that's basically of size equal to the sum of the previous 2. So if you look at the probability of running a paper with 100 citations, it's 4.5-fold higher if it's written in a team, right? So it's a 4.5 times increase. This isn't a small study. This is 22 million papers or something, right? And what Brian will say in a nutshell is, one of those comes from basically, doing important stuff. One of those comes from atypical combinations, and 2 comes from doing important stuff that's combining atypical stuff. Now, Dashan Wang is at Northwestern, just unpacked this more, and what he found, which is fascinating, this relates to Bob's point, is that breakthrough ideas like Kahneman and Tversky come from groups of like 2, 3, 4 people who are working in different disciplines, right? Because you've got to have, if you go back to the thing I talked about at the end, a deep engagement in the ideas. Once those things get pushed forward, then it's bigger teams of like 10 or 12, 7, 8, 9, that they call developing papers. And so the developing papers now, especially as there's more data, might require a biostatistician, a statistician, a mathematical economist. Like Carl Simon works on all sorts of papers, or someone like Mark Newman in networks, right? Rabbi Jacobs, who we just started, does networks. You may bring someone like that in who doesn't know the field but has a skill, right? An analytical technical skill. And so there's really, in some sense, kind of two stories, right? One is the breakthroughs come from these atypical combinations that are new and deep in smaller teams, and then the developing comes from sort of multiplicity of tools and frames and models to make sense of things across groups. But no, absolutely, like the value, as problems become harder, it's just going to be too big for this pumpkin, right? And so we're going to need lots of people, and we're going to need lots of people who are trained differently. And the value is going to come from teams, and just can we learn to make those teams bigger and better?
I was wondering, sorry,
Yeah.
that maybe it's exposed to more disciplines and more disciplines quote those papers or something.
Yeah, and the other thing that comes out of this, ethnic diversity, this is by Richard Freeman, gives you like a 5% bonus. Hold everything else constant and just change like the ethnic diversity of one of the authors, 5% bonus. The really harrowing piece of results as a social scientist, if I were just to work with someone from Ohio State, 10% bonus. Not going to happen, hypothetically, 10% bonus.
And on that note, let's thank Dr. Page for his DUP lecture. Thank you. I'd now like to thank you for having attended this Distinguished University Professor Symposium and invite you to attend the reception, which is just outside the doors behind me. Thank you very much, and good afternoon.